Hey biology students! In this lecture video, we're going to walk through the steps of adaptive immunity part 2, the humoral response. And this is the response that involves the B lymphocytes, the B cells. So in the previous lecture video, we described the cell-mediated response of adaptive immunity, where the T cells become activated. So quick review on that. So in the previous lecture video, we described what happens in colds and flus, as well as the coronavirus, how viral particles enter through the nose or the mouth as droplets, and then the viral particles, if they get past first line of defense and initiate second line of defense, where we get inflammation and we get activation of phagocytes and fever response, and all of that is going on. And so some of those antigen presenting cells migrate to lymph nodes, where we get activation of helper T cells, and then ultimately activation of cytotoxic T cells. And we drew this summary map in the last video, where we showed this process of activation, ultimately leading to cell death. These would be, of course, our cells that are dying, the target cells in the case of viral infections, where the attack of the virus is intracellular, meaning the virus goes inside of the cell, replicates inside of the cell. That becomes a viral cloning factory. That is not a good thing. We need the viral cloning factory to be shut down and destroyed, and that's going to be accomplished by the cytotoxic T cells. So here in this diagram, we showed that a cytotoxic T cell is releasing perforins, a type of cytotoxin, a toxic protein that kills the cell, and that cell dies. And recall that this cytotoxic T cell has made millions of copies of itself. It's gone through a process called clonal selection. And in addition, the helper T cell that matches this particular antigen also has gone through clonal selection, and this helper T cell is providing the second signal needed to activate this cytotoxic T cell, because the cytotoxic T cell has to receive an interleukin signal from a corresponding helper T cell, and then it also has to bind to antigen complex with MHC1 in order to initiate this response of killing the target cell. So cytotoxic T cell has been activated, and now we want to talk about how the B cells fit into this story. And so what happens is we are shutting down these cells that are replicating new virus. However, are there still viral particles in circulation in the body at this point? Yes, there are. So there is a part 2 to this adaptive response, because we still have viral particles in circulation, and the T cells, they cannot directly kill virus. They can only kill cells that are infected with virus. So they cannot go on a direct kill of the virus. Make sure you understand that. So the only way that that works would be if we initiate the B cells. So we need the B cells to respond to what we are calling here, which is it's free antigen, basically. It's viral particles that are in circulation, maybe in the blood or in the tissues or in the lymphatic vessels. They are still in circulation in a very large quantity. Now we're shutting down the shop of replicating them, so that's the job of the cytotoxic T cells, but we still have free antigen in circulation, and that's going to need to be eliminated, and that's the job of the B cells. So if we take a look at the map here, if you want to add to this map, then this helper T cell that became activated in the previous video, we talked about finding, the antigen is sort of like the wanted poster, and the antigen presenting cell creates this wanted poster presenting the antigen. Do you recognize my antigen? Do you recognize my antigen? Until finally the helper T cell that recognizes that antigen is found in the lymph nodes and then begins cloning itself and going through clonal selection. Well, this helper T cell has to also activate a B cell. So B cells have to become activated, and then when B cells become activated, we see that B cell differentiate into plasma cells, and those plasma cells create antibody, and then some of those plasma cells also become memory B cells. So let's tell that story in a little bit more detail. So let's talk about a B cell and what that B cell looks like. So B lymphocytes look a lot like T lymphocytes with a large nucleus in the center. However, the B lymphocytes do not have the same type of receptor as the T lymphocytes. The B lymphocytes have antibody receptors. Now we do call these B cell receptors, but they are Y-shaped protein receptors. And there are many millions of different types of B cells that exist in the body as well. So real quick, let's just draw in a couple more over here. We'll put in a couple more B cells. So we'll here's another B cell, here's another B cell. And let's just go ahead and make their B cell receptors a different color. So we'll make the B cell receptors over here red. And that's because they recognize a different antigen than this B cell with the green antibody receptor. And we'll make this one over here have a blue receptor. So each of these B cells, just like the T cells, recognize a specific antigen, and they have to have the correct receptor to match that particular antigen. So what's the antigen? Well, in our story, why not make a coronavirus, right? It's on everybody's mind, so why not? So we've been telling his story, and coronavirus is it's a little spiky virus. Okay, it has this little crown of spikes. That's why it's called a coronavirus. Corona means crown. Here it is. So here's our little virus, and that is the target. So it turns out that only one of this group of B cells would recognize that virus. And I've gone with a color coding theme here. So the one in the middle, this B cell here has the green B cell receptor that recognizes this particular virus. Okay, so this virus can come and actually bind to this B cell receptor, and it actually binds at one of the tips of the Y, because those are the antigen binding sites of this receptor. So the virus binds there. Now, what happens? Well, the B cell actually will engulf this virus. So this binding event will trigger the B cell to endocytose the virus. And so it'll endocytose it, bring it into the cell in a little vesicle, and it will bring in the virus and it will digest the virus. So it'll break apart the virus inside of a phagolysosome. And then what it'll do is it will display the virus on the surface, complexed to an MHC class 2 receptor. So this is an MHC2 receptor that is now bound to viral antigen. If you recall, there actually are 3 types of antigen presenting cells in the immune system. There are macrophages that are the king of the phagocytes, and they become antigen presenting cells when they engulf pathogens. There are dendritic cells, that would be the second type of antigen presenting cell. Now, both macrophages and dendritic cells, their main job is really to be a phagocyte and to be an antigen presenting cell. So actually we refer to macrophages and dendritic cells as professional antigen presenting cells, meaning that is their profession, that is their job, to be an antigen presenting cell. So B cells are the third type of cell that can present antigen in the immune system, but we don't call it a professional antigen presenting cell because that's not its main job. You're going to see what its main job is in a second here. But do note that it does process antigen and it does present that antigen complex 2. It's MHC2. It's the third type of cell that carries the MHC2 receptor. So it's complexing that. And so who recognizes MHC2 complex 2 viral antigen? Well, actually, let me draw another one over here just because I need some more space. So we'll say that it's complexed over here as well with an MHC2, complex 2 antigen. Okay, so this B cell that is now displaying antigen, and we'll actually go ahead and call it an APC, but it's not a professional APC because that's not its main job. The B cell here, if it binds to antigen in this way and presents antigen in this way, it will actually just sit there and do nothing until it receives a second signal. So there is a second signal that's critical to activate this particular B cell in this response. So who is going to activate it? Who recognizes, what immune cell, if we learned, recognizes MHC2 antigen complexes? That's right, helper T cells. So the second signal for B cell activation has to come from a helper T cell. So remember this, oops, where is it now? This diagram here, where in the diagram I'm showing you that this B cell is becoming activated by a helper T cell. But really it's two things that have to happen. Two things have to happen. One, this particular B cell is in circulation in the blood, and it has to become activated by binding antigen, complexing that antigen to MHC2, but it's just going to sit there and wait until it gets the second signal. So the second signal comes from our helper T cell. So here will be our helper T cell, our TH cell. Remember, this is a TH cell that had to be found. This was Waldo, had to be found. We had to have him activated, and he has the matching TCR. So he has a TCR that's also color-coded here to show that it matches this antigen. So he has the TCR that recognizes this particular antigen. So this is the TCR. And it has to bind to the MHC2 complex of the B cell. And that's what T helper cells do. So they bind here. And then once that helper T cell binds, it secretes an interleukin signal. So here's that second signal that the B cell needs in order to get the instructions that it needs. These chemical signals introduce a signal mechanism, a cascade reaction inside of the B cell that tells it, okay, you're activated. You located the pathogen, you located the bad guy. Now you're getting a reinforced signal from the helper T cell that now you need to divide. And so this B cell will respond to the interleukin signal by cloning itself. So it's going to make many identical copies of itself. So it's cloning itself. This is also part of that theory of clonal selection. So there's really three lymphocytes that we describe as undergoing this process of clonal selection in adaptive immunity. And it would be the helper T cell, the cytotoxic T cell, and the B cell. So all of those that go through a clonal selection process where they're activated only because they recognize the specific antigen involved in this particular infection. So notice these B cells that are hanging out over here in the wings, they are not activated in this particular infection because they carry unique antigen receptors, the B cell receptors here that bind to different antigens, not the antigen that we're interested in. The specific antigen here is going to be recognized by this B cell here and this particular T cell. So then what happens to our B cell when he's activated? Okay, so we get B cell activation. So we get B cell activation. Now what happens? Well, the B cell becomes a plasma cell. And remember, we show it looking like this because it has a whole lot of endoplasmic reticulum inside of it, meaning that it has a lot of rough ER. It has a whole lot of machinery to make the corresponding antibody that we need for this response. So it's going to start synthesizing antibody protein inside of it, inside of the plasma B cell, and then it's going to secrete the antibody. So we end up with antibody in circulation in the blood. Millions and millions of these antibodies go into circulation. So secretes antibodies. And remember, let's go back to this diagram. Remember this process here of this viral infection. In order to wipe out the viral infection, you need activation of both a cell-mediated response. So we need those cytotoxic T cells acting like hound dogs and sniffing out where the virus is replicating, and then lysing those cells, those virally infected cells. But we also have free antigen floating around in the cell fluids as well as in the blood, and these guys are looking for target cells, and we need to eliminate those. So the B cell response is necessary as well. In order to wipe out a viral infection effectively, we need both branches of adaptive immunity. We need the B cell response as well as the T cell response in full force. So now free antigen. Now here's the beauty of the humoral response, of the B cell response. The beauty of that is that they recognize free antigen due to the antibody response. So B cells don't go on the attack themselves. B cells differentiate to plasma cells, and then those plasma cells produce antibodies. So here comes the antibody. What's the antibody going to do? Well, we're going to do a lecture on that in the next video because there's a lot of things it does actually. But one of the things it does, the antibody will coat the outside of the virus, and that's going to neutralize the virus. That's going to prevent the virus from being able to attach to any further cells. So this free antigen has now been neutralized by antibody, eliminating that virus from binding to any more cells. So we need to coat, and we'll coat this guy too. We'll just tackle him down, tackle him with these antibodies that are very specific to the particular antigen that's involved in this infection. And so now we have a response that is wiping out cells that are breeding new virus, so no more new viruses being made, and any viral particles that are in circulation in the blood or in the body fluids are going to be bound by antibody and wiped out. So that is the way to effectively clear an infection caused by virus. Viruses in particular need both branches of adaptive immunity. In bacterial infections, a lot of times only the humoral response is needed because the humoral response, the B cell response, is in response to free antigen. So for bacterial cells that are in circulation, those can be neutralized by antibody. A lot of times the bacteria don't go inside of the cell, so we don't really need a cytotoxic T cell response, but hopefully you understand that viruses require both types of adaptive immunity. I also have a visual animation of this that we can take a look at together. So in this picture, they're going to show you antigen presentation. So they're going to show, hey, look, the virus, little green spiky guy, just like my pictures. That's coincidental. Okay, so they're showing us the engulfment of the virus by a macrophage. And then the phagolysosome, the phagocytosis, and then it's showing us that here comes that MHC2 receptor that binds to some of the viral antigens, and now it's an antigen presenting cell. So you know that story. And then it goes to the lymph nodes and interacts with the helper T cell that happens to have the matching TCR receptor, and then the helper T cell becomes activated, and then one of the things helper T cells do is they bind to the MHC2 complex on the B cells that have recognized the antigen, and then when that happens, that produces an interleukin signal that stimulates these B cells to divide and become plasma cells and secrete the antibody. There goes the antibody. All these little Y-shaped proteins get circulated through the blood, and they seek out like a magnet. Think of antibodies like a magnet. Okay, they're acting like a magnet to seek out that particular antigen and prevent it from causing any further damage. So we're going to do the next video on more details about antibodies. However, I'd like you to leave you with this diagram here. So this is actually showing us the antibody response, which again is the humoral response. And the name humoral, the reason why the B cell response is referred to as humoral immune response is actually for historic reasons. As when people were first trying to understand biology, there was a lot of incorrect information. And one of the incorrect information, that sort of a myth, was that your humors or your personality comes from things in your blood. And so your humor, your personality is blood-borne. Anyways, that has sort of stuck. And so when we use the word humoral, it just means blood. So what, we're talking about immune response that's basically all about what's happening in the blood. And that's because antibodies get secreted into the blood. So this is showing us something called the primary and secondary response graph. This is a graph you should be able to understand and interpret. So on the y-axis, it's showing us antibody titer. Basically, that's concentration of antibody. And then on the x-axis, we're looking at time. And that's highly variable, days, weeks, whatever. But it's showing us that at time 0 here, at antibody level 0, is your primary exposure to the antigen. This is the first time you get sick, okay, or the first exposure to this particular pathogen. And then there is a time where there's no antibody made. Notice it's a flat line here. It's called the inductive period. Why is there no antibody made for a long time, maybe up to 10 days? Well, it's because there's a lengthy process going on for antigen presentation to occur. Okay, so remember, there's always the first and second line of defense first before we launch the third line of defense, before we start getting specific with the antigen. So all those things have to happen. We get inflammation and we get antigen presentation, and then we have that antigen presenting macrophage has to go to the lymph nodes and seek out Waldo and find Waldo, and Waldo, of course, being the helper T cell, and then activating the helper T cell. And this all takes time. So that's why there's this flat line for a while where all of this antigen presentation recognition process has to occur. Finally, the B cells get activated, and then we start getting antibody. We get this little peak of antibody, and then we get a decrease in antibody, and that's just because the infection is over. So this will effectively eliminate this particular pathogen. Does it always eliminate the pathogen? No, it doesn't. Sometimes, even with an immune response, the pathogen is able to overwhelm the body, and then that can cause death. But let's be positive and talk about when you recover. So you recover from this infection, and the antibody response decreases because the infection's over. We don't need to make a whole bunch of antibody if there's no more pathogen in circulation, right? So that gets eliminated, and the memory cells are what are left behind. After the infection, we have these B cells that are going to patrol the body in the event that that same antigen is ever introduced. So then they say, okay, X amount of time later, there's a gap in this curve because they're saying, who knows if you'll ever even be exposed a second time to that antigen. But if you are exposed to that antigen, look what happens. This is what's called the secondary immune response. So primary immune response is what we just saw here. Secondary immune response is what happens if you get exposed to the same antigen. Okay, so the same antigen. Not a different antigen, but the same antigen. Same antigen again, what happens? Notice there's not a long inductive period. There's a very short inductive period. And that's because of these guys. The memory B cells are on patrol, and they recognize that antigen quickly and start making a whole lot of antibody. Way more than actually you did originally in the first time you were sick. Second time you're exposed to that antigen, you're actually probably not even going to get sick, meaning you're not going to have symptoms. You're not going to get inflammation and runny nose and cough and everything that happens in the inductive period over here where the second line of defense has to labor through the battle. You're not going to get any of that over here. You're going to skip all of that and it's just going to right to antibody. It's going to go to a boatload of antibody and then that antibody is just going to clear that infection for you. And then slowly that antibody will decrease as that infection is cleared out of the body, and you may not even have recognition of that happening. And that is remarkable. That is the beauty of having either natural immunity to a specific pathogen or to have been vaccinated. So if you've been vaccinated against a specific antigen, you have memory cells that have been created from the vaccination process, and then if you are exposed to that particular antigen a second time, then that memory B response is a very powerful response and eliminates the pathogen before you even are aware that you are sick. Okay, so that's the humoral immune response, and we will talk more about the antibodies in particular in the next video.